So the first thing you want to do is to update your toolbox. So you can a toolbox quick installation. So you can copy everything here in this box to reinstall the package, or you can just copy the unzip. This single line should also do the same work. So that's what I'm doing. I copy this and put in unzip. So this unzip function will download this zip file and unzip here. So I have my updated version.
So if I install in the lab, how do I know
which version? So I should have a version number right now. Yeah, so I should have this version number right now in the toolbox.
So maybe just every time use the before we
Yeah, yeah, because it's I always update it. So yeah, you just I have a labeled version number on the top right now. The reason I want to update it because remember last week we had a bug in highly variable gene output, but now I fixed that bug, so we can continue with the highly variable gene analysis, and then we move to other topics. Okay. So if you are ready, we are going to use example data again to do the highly variable gene analysis. So this highly variable gene analysis is to try to identify highly variable genes from one population. There's no comparison, just from one group of cells, and also require these cells to be same type and the same states, so this homogeneous population. So from there, and we will check which gene show this highly variable pattern compared to other genes, basically. And those genes, as we described, are most likely to be very important to reflect the biological function about this population of cells. That's why valuable. So what we can do is here to use this updated version. If you use older version, you still get the same bug. Okay. I want to do the simulation first. Okay. Let's just simulate a data. The simulation is based on our statistic understanding about those distribution of those counts. Okay, I will simulate 3,000 genes in 5,000 cells. So I assume this is a homogeneous population, so I can apply highly variable gene analysis to see what other highly variable genes among those simulated data set. You can directly click this. This is a highly variable gene analysis. Or if you use manual, you can use this. This should have highly variable gene. Yeah, so the button and this menu item are the same, okay? So if I click this, there's a description about this annotation, the method. Then you click continue. This will ask you, analysis will apply to all the 5,000 cells here, okay? Just make sure they're homogeneous. Then we can use spleen fit. This method is recommended.
So last time I think I tried both methods. They give you the different.
Yeah, that's a very different implementation. So I suggest to use this 3D, this by default. So what you can see from the simulated data, there's no even a single gene jump out from this regression curve. They fit all the expectation nicely. There's no noise, there's no biological variability among simulated data. So each point here is a gene. Because this is simulated data, so I don't have a gene name, I just have G1, G2, G3. So you cannot do any function annotation with those genes. But what I want to show you is simulated data has no variability. There's no gene jump out from this regression curve. And on your right side, this is an expression profile of these genes. You can have, for example, this one have quite a lot of zeros. Only 10% of cells express this gene among 5,000. But their mean and CV are all expected. You can explain with relationship with other genes. So that's the simulated data. Now, we can start to use example data set. Okay, you load example, process the example data. So this is a real data set from a mouse. Now the highly variable gene expression analysis require a homogeneous population of cells. So I'm going to select one of the group rather than apply highly variable gene analysis apply to all the cells. Doesn't make sense, because in that way we probably only get those marker genes expressed in one cell type but not another. So that's give you the variability switch on off among cell populations. But what we really want is to, among this homogeneous population of cell, what the highly variable genes are in there. So I select this and see I want to delete all the rest. Brush the cell only. I select brush cells and then delete unselected. Okay, if you do this, you end up with the population with 937, the one you brushed is selected. The color changed, but they're same cells. Okay. Oh, yeah. So I can show that again. Okay, I want to keep the cells I selected, so this group. So I brush them. And then click this button here, see delete, brush the cell. But I will delete unselected. Brush cell only, and delete unselected. Now in this population, you want to apply QC again, because there are some genes may be completely non-expressed, but they are still there. So I want to remove those genes before apply highly variable gene analysis. Okay. So now we can click this button here to get highly variable gene out of this population of cells using spleen fit method. Yeah, we can start to see some of the genes jump out, but not many.
Oh, the first one that pops out is a red blood cell?
Oh, red blood cell, we should, yes. It's kind of a contamination, okay? Yeah, this is hemoglobin. You can change this transparent level to look at the gene dots. You can highlight. So this jump out. This is a contamination caused by the red blood cell contamination. So both of them. Those ones start to show the genes maybe have really meaning of function. This is insulin. So the population I select probably is a beta cell. I don't know exactly the cell type. Let's try the cell type. This endocrine cell. Okay, with this, we can see I want to select top highly variable genes. So top 100. So this method will help you to select those genes, deviate far away from the regression curve. Yeah, as I said, these two are contamination, the hemoglobin genes, the top two. But the rest of them can help you to understand the function. So once you select those genes, you can save this gene name and save into a table for the downstream. You can send those genes directly to enricher. So if you click this button here again, this gene name will be sent to a website. So the website, including those genes, okay, 100 genes you selected. I just keep those contamination genes, but maybe I just remove them. You can submit to this website to do the enrichment analysis. I remove 3 genes, so only have 97. I can look at as a gene set what they are, their function are. So for this group of cell, endocrine cells, and we have those top highly variable genes, including those response to unfolded protein, some heat shock protein, regulation of including body. Yeah, so that this molecular function, including, yeah, because those are the hemoglobin genes. If you look at this plot here, to view hemoglobin gene expression among those cells, you will see this mouse. There are some of those cells contains hemoglobin genes. Normally they shouldn't. Okay, those from the contamination from blood cell. Yeah. Yeah, you can delete those. Clearly not meaningful. Yeah. Oh, if you want to check the contamination among your cells, you can click this view and see, okay, hemoglobin gene expression. So all the hemoglobin subunit genes will be concatenated, put together, and if you have a cell, have a lot of those genes, that cell could be red blood cell. And because this gene is so specially restricted expression in highly red blood cells, so if you have that gene in there, it's highly variable. It's only expressed in 1 or 2 cells, but 1,000 times higher than others. So the algorithm will pick it up as a highly variable genes, but you know it's not relevant to the cell type you are working with. Yeah, so let's see. So what I can, yeah, I haven't developed a function to delete it, but if you know this cell, and then you probably can highlight them and delete those two cells. Yeah, so you can highlight and delete those two. Yeah, so after that, then view hemoglobin again. I still have one. Yeah, yeah. But those low levels should be fine. It's very small, yeah. But this one, if we can delete that, it will be good. That's one in the middle. But if you run highly variable gene analysis with this population, then I hope the hemoglobin gene will be not included as results. Yeah, so they should be gone. Yeah, it's still here. There's one still here. This is contributed by just one cell, right? Okay, any questions? Yeah. If a gene you are interested in is not in here, yeah, that's because that gene not expressed in this population of cells. So the filter, the QC filter, will remove that gene. No, this is just random, the cell 1 to here, the 900 cell. If you do on the same data set, it will be the same index. Index order will not change. Mm-hmm. If you have a expression, see, okay, this gene only expressed in the top 600, but not in the other 300 cells, that's clearly different, then that means the population you're working on have a structure. Yeah, that's not a good indication for the highly variable gene analysis. Highly variable gene analysis assume all the cells are the same. Yeah, not like a structure, have a half this, half that. Mm-hmm. In here, I select top 200 genes to see whether I can get some other annotation, some receptor inhibitor activity, ligand receptor, double strand. Yeah, so there's some of this. If you know the biology behind those genes, maybe they can help you to understand the cell population themselves. Okay. Okay, so we are going to move to the next topics here, okay? This is highly variable gene from last week, remaining issue. So now we are going to look at SVD function. You can close all. So let's construct a small example matrix. You can do this, okay? You cannot do eigen decomposition with this because this is not a squared matrix. But you can run SVD with this. So you will get U and S and a V. So you decompose a matrix into three matrices, okay? S may not be in the matrix form, but you can use the diagonal, the function, to make it a matrix. So let's see, this U will be 2 by 2, okay? V is 3 by 3. S is a vector for singular value, but if you put singular values in this diagonal position, so S is a matrix. So your original is this, but you decompose into the direction of 2, 2, 3, 3, and also this linked matrix here. How can you put this back? You can use U times S times V transposed V, and you recreate your original matrix. So it's a decomposition and then put back by the matrices multiplication. So the question is why we bother to do this. The reason is after you decompose, you look at your matrix U and V and this S, the values, they will tell you the relative this component's contribution to explain the original matrix. The most important signal is from S. So S tells you this is a 9.5 and another component is only 0.7. So this top component can explain much more of the value difference in your A matrix than the second component. So if you get this idea and you want to keep only one component rather than construct the whole thing, you can see I want to select the U and only the first component. So U1, okay, from U, you select only one column here. And S, you only select 1, 1. V1, you select V, only the, I don't know the first row or first column. Let's try first row. You have in your matrix, you have two components, but now you only use one. And you can still construct majority of this structure. I hope this works. Yeah, so I need to know how to construct this. So if I use B column, yeah, so this is, so I use the first components to reconstruct. This is not perfect because I lost the information from the contribution from second component, but most of those values have been, original was 1, 2, 3, 4, 5, 6. So it's off a little bit, but the majority of those have been reconstructed. Does this make any sense? No, still not. We have another 2 examples, I hope, at the end. Okay. Yeah. Yeah, so let me do that, the code again, okay, from beginning. So I have A here, right? I decompose them into USV, but I only use U1, only one from S, V1. And then I reconstruct original A by doing this. Not the whole matrix, but reconstruct most of the values close enough. This is a small example. If you have a big input A, and then you select, for example, only top 3, you can reconstruct majority of them. That's the bigger advance. You compress information and extract the most useful information. Yeah.
So if you're looking at like your own data set and you have, like, I think the number you got was 9 because that's the greatest contribution, how do you know what factor that is in your data set if you're just looking at the matrix?
Yeah, so this S matrix, the algorithm have already helped you to reorder. The first one is highest, the second one second. So you can keep the top K, whatever number you want to keep. Yeah, everything is already ordered internally by the algorithm. So I have a good examples for next one. It's view from the slide here. Okay, this slide is from so let me yeah, so I want you to type this matrix in as your A and use SVD to see whether you can get those matrices. Okay, you know how to type this in, okay, with A as A. So this example is slightly bigger than the 1, 2, 3, 4, 5 to the original, the one I just used. So same thing, and A is first row 1, 1, 1, 0, 0. Okay. You put a semicolon here to change the row definition, you can put the 3 dots here in order to continue. And 2, 2, 2, change 0. And then close. So you have this A matrix, this 7 by 5, okay, 7 rows, 5 columns. There is a function to help you to visualize the values. This is called image SC, and you put the matrix in here. So this will create a so-called pseudo color image, and to have a lot of box, and each box corresponds to one of the values here, and based on the value, and they will give you a color based on the color map you use. So 0 here will give all this dark blue, and high value will be yellow, okay, and they're the gradients. So this gives you a quick view about the matrix you have here. It should work with hundreds by hundreds rows columns, but if you have thousands by thousands, then the resolution normally not enough to show you this. But for this small example, we can have a quick idea of this structure in the values of this matrix. Yeah, questions, Anastasia? Mm-hmm. Yeah, type that in and then use image SC to see that. You can change the color map if you want. You didn't get. It should be somewhere. It's behind here. Yeah, you can shut this down. Do that again. Yeah. It should come out. Yeah, yeah. You just superimpose onto that. Yeah, overlap. Yeah. No problem. Yeah, so this color can be customized, okay? There this color. If you select edit the color map, you can select a different color map. By default, this is called a perula. But if you select the copper, so this gives you the this gradients from black to light copper side, the jet. Okay, so this gave you different color map you can use. To indicate the values. So some of those color map are for the continuous changes like this. You have gradients from one color to another. Some of those will be discrete, like this lines. This will be given alternate color to indicate the difference of a category difference. So it's not a continuous change. So this will be not a good options to view the values, the greater or smaller values in your plot. All right, any questions? So now we are going to decompose this A matrix, exactly the same function as VD. Now the U matrix should be 7 by 7 because you have 7 rows, and the V matrix should be 5 by 5. You have 5 columns, so one is from that direction and then another from another direction. S should be 7 by 5, and only the diagonal position S have values. So if you look at these values here in the S matrix, the first one is 9.6. The second one also contributes substantially, 5.2. But you move to the third one. The third one is the 10 to the power negative 16, so it's almost 0. So the rest of them even smaller. So there's a clear cut that the first 2 components contribute to explain the variability among your matrix, but not the rest of them. So if you don't do anything, you just use the whole decomposed matrix to reconstruct A, you get the same values, okay? But you know, only top 2 contribute the most. So we can see, okay, I use top 2 instead of U2. So it's from 1 and 2. Okay, I use only 2 columns from U. Only 2. So B1, only 2 from so I just keep 2 components from each U, S, and V rather than use the whole. I can reconstruct to reconstruct A. So A reconstructed. Yeah, you see, I get rid of all the small components and only use top two. I completely reconstruct the matrix because this matrix is highly structured. So only two components actually contribute to this. So that's why in this example, this is like U, and that's S, that's V, but you only use two of them rather than the full matrices. So by look at the values here, and also you start to recognize those samples and come from the first components. Those samples come from that component. So you start to have some idea about this, where the those variability among samples come from the concept map. So this matrix here in our examples is U2. This U2 should be so this is almost zero here, okay? Those are the values. And your V2 is here. In the example is here, okay? There are negative sign in each of those values, but they all canceled out based on the direction of your algorithm. There's nothing here. Any questions for this second example? So if I use images to do this reconstructed same picture, you can also change the color map, use command line. So here the way is to do color map, and then you have winter, then change to that. If you have a gray, it will change this gray scale, okay? Default. Question? The V? Yeah. So by default, this, as SVD algorithm return a transposed V to give you. So when you to do the reconstruction, you have to rotate it. Yeah, to do. It is by default. This from engineering, and why they gave this transformed. The function gave you a transposed V. Okay, we will look at the third example of SVD. Clear your workspace. So I have trouble to open my Teams here. I don't know what's going on, but I want you point to this page here. I should have the link in Teams. You just click that. In this case, we want to use SVD to do an image compression. So first, I will read this image into a matrix A. Okay, so this function just read an image in the JPEG format and into a matrix. This matrix contains 3 layers in different 3 colors. Yep. To find the page, so the page is called here, bigger font. Mm-hmm. I hope. Is that okay? Good. Yeah, so this read that into a 3D matrix. 3D matrix is called tensor. The reason is we have a color image. Okay, 3 layers and 1 each for the different channels of colors. We can convert that into 2D and by getting rid of the color. So this function will change the color into non-color. So remove the color, just use a scale. So yeah, so now this A becomes a 2D matrix. Yeah.
I was going to say, if you just, whenever you open it on that one,
Mm-hmm.
if you just hit copy command and then paste it here, then it opens the live script, so then you can just like execute or evaluate it.
Yeah, that's good. Yeah.
So that you don't have to go back and forth.
Yeah, this, okay. Yeah. Okay, that's an easy way for me, but maybe not an easy way. Yeah, I want to slowly copy and so people can follow. Yeah, good. So this convert this original 3D matrix into 2D. So now we have this 480 by 640. So you can view this by image show. Okay, that's the function here. Okay. So the values in this matrix just gave you the density of whatever called scaled grayscale values. I believe you have, if you have a 0 here, you should have a black dot. If you have 250, it will be white. So this is scale from white to dark, and then different levels of the gray. So we have this count matrix. It's very much like the single cell data, right? So we can do SVD for this matrix A. This matrix A, because they are integers, so the data type was converted into so-called unsigned integer, but we have to make them double, D-O-U-B-O, double A. Okay, just change the internal the data type, not the value itself. But the original one is for the compression to save the memory or hard drive. But to run the function of SVD, we need to have a double data type. So you have this original, and then we can use this function to do this so-called sketching. This function will automatically help you to select, how many components you want to use in order to keep the original matrix in what kind of level of the precision, accuracy. If you select more components, you will reconstruct the original picture almost perfectly. But if you select only top 2, you will lose a lot of details in this original picture. So this SVD sketch, if you give this cutoff, it will automatically help you to select the level of the reconstruction. So if I copy this and then reconstruct, so use U1, S1, V1 reconstruct, and then make this image back. I should have okay. So looks to me it's almost the same as original. Yep. This line, that's the end. Before I copy this, yeah. So if I change this, okay, let's come back.
That's just an example to get a higher resolution
Okay,
instead of lower resolution.
okay.
And they have a spline version on the very bottom too.
Mm-hmm. Okay, so this is to go different directions. So this you reconstruct high resolution picture. Okay, that's interesting. So you can have a different technique to interpolate the data to make a high resolution even. Okay, that's interesting. So I'm going to try the different cutoff. I have never tried this. I just give it a try, see whether I will construct a better or worse resolution using different cutoff. So it looks like this is worse if I set different cutoffs for the SVD sketch. Here only use 48 components to reconstruct the original picture. Okay, so this is a
So the decomposition will increase the process time?
decomposition.
Like for real data, let's say single cell, you make it a smaller matrix, right?
Yeah, you make
the matrix will be smaller, so
yeah.
that will increase the whole process time, or?
Yeah, you can use this as a image compression. Rather than send the real picture, you can send those components to another computer, and another computer will construct the image from the matrix multiplication. Yeah. Okay, it's time for 5 minutes break. Okay. Yeah, Victoria, so this increased image resolution, you have to use this called interpolation. So if you have a missing data in between of two points, you try to fill it up with some technique. Mm-hmm.
So it's kind of like the opposite of like
Yeah.
how I guess Androids have that feature on their phone where you can remove something from the background. Like if you take a picture
Yeah.
and somebody's in the background, you just like take them out.
No, it's still different.
And then it like fills stuff in.
No, different. This is you increase resolution by interpolate the point which in the original points, but between, so you fill up in some new data. Mm-hmm. Okay, see you in 5. Oh, yeah. Question? You mean the video? Yeah. Oh, you want to download MP4 file somehow? Yeah. Let me find a way to send you this Google link, something like that. Put the MP4 file there. Yeah. The channels for. Oh, yeah, I had YouTube channel, but it take time to prepare to upload into there. Yeah. But I have, yeah, I will give you the YouTube channel link later. Okay. Yeah, you have everything. All right. Yeah. So this example is just you the original picture, and also you use different levels of the reconstruction, the components. And when you use only 15 components, you get a low resolution picture compared to the original. You can increase so-called rank. Okay. Rank means how many components you used in the reconstructions. Yeah. I think so. I encourage it within student discussion, for sure. Don't worry, it's recorded. Yes. So you can use this to use less components in order to construct the original matrix to a certain level of the quality. And then the information you used is much less than original matrix. Okay? So that's why this is a compression. If you send a huge image across internet to another computer, you can rather send those components and give to that computer to let the computer to do multiplication of matrices to reconstruct original information. So the data you transferred between PCs, between computers, will be much less than your original picture. So this is kind of a compression. We can also use this technique. This is called low rank SVD or SVD sketching to remove the noise. If you have original picture contains a lot of noise, not in this case. And then just imagine you have a lot of noise here. But after you do this, you actually smooth out the signals. And this looks more smooth compared to the original, if your original is a noise picture. So that's probably what we want to do for single cell data. Single cell data is so many variabilities. And here, if you do the smoothing, and then they look more continuous. Yeah.
I'm just thinking, how do we know when is too much movement? Like, how do we know where the upper limit for movement?
Yeah, we don't know. We don't know which level you want to use. The hyperparameter, the cutoff, you can set. Let algorithm to decide for you. You can try different outcomes. So okay, at this level, you are okay with that. For example, you have a super high resolution of picture, but in this level, you cannot tell by eye the difference, then you just keep that level. Yeah. We have no idea when we stop to at which level you do this denoise. If you over smooth your data, maybe you looks good, but you lost those biological variability. Yeah, yeah. We don't know. This is okay. Another example. we in the channel we have, so I go through this. In the channel we have this zipped file, and this zipped file contains ICA example. We can all download this. Let's see. How can I download this? Okay. You click here and download this zipped file. Then found it in your download folder here. Okay. And then right click to extract this folder, this zipped file, and put in the folder. And then you copy the path of this folder. And then come back to MATLAB. We are going to close all, then CD to that folder. So that's the folder I unzipped. The example I download. So I change my working folder to the place where I unzipped my zipped file. Do you want me to show you that again, the procedure? So in the channel, you have this zipped file, and then you double click it, and from these three dots you download this zipped file into your download folder in your local computer. But this is a zipped file. We need to open it to extract the files in there. So you have to be in this download folder and extract all. So to make a new folder, extracted. And you have to locate this folder and copy the path to the folder and come back to MATLAB to do the CD. CD means change the directory, change the folder to the place where you unzipped your file. So at this moment, if you can see this unzipped folder, and also you can run this ICA demonstration. So ICA is to separate signal into their independent source. So in this example, we have three different sources of the sound. This one, okay, then you have
long list of American fast foods that have success.
Okay, and the third, so you have three sources of sound, and then this example will first mix the sound, three of them in different proportion, and then you have this mixed one.
Long list of American fast foods that have successfully made.
And then you apply ICA, you should be able to separate the 3 signals into independent channels. And then you have another mix. And you provide this 3 mixture. One is stronger in one of the source, another is stronger in another. So 3 input mixture signal give to the algorithm. Algorithm should be able to reconstruct the original. So this is reconstruct the second. This reconstruct.
Long list of American fast foods that have.
Yeah. So using different algorithm, ICA. Also, reconstruct the 3.
Long list of American fast food.
Another reconstruction.
Long list of American fast food.
This algorithm is not as good as this original, so they still have some mixture.
Long list of American fast foods that have. Long list of American fast foods.
Yeah, so this is a demonstration of the possible. I'm not going to spend time on the details, but all the source code is here. All this soundtrack files in the audio folder. The signal was read into and then mixed in this, and then use this so-called fast ICA to decompose. Questions? Well, okay. Yeah.
So would this be like in medical imaging terms, I guess it would be like high pass or low pass filter because you're
Mm-hmm.
you're mixing the signals and then I guess you're trying to separate them out.
Mm-hmm. Mm-hmm. Yeah, so when you see high pass, low pass, it could be based on the frequency, and then you try to filter out some high frequency pitch noise. Yeah, slightly different from this. If you have the cutoff where the frequency, there's some signal processing algorithm certainly. The SVD? No, it's different from SVD. It's very, very different from SVD itself. Yeah. The purpose, the SVD, you can get rid of the noise, but the data type for the SVD is not this continuous, this long time series signal. Yeah, so not directly related. Mm-hmm. Yeah, I don't have a good examples for using ICA in single cell data. But if you have some good ideas, say, how can I use ICA to analyze single cell data? That will be a good project presentation for your final. Okay. All right, I will let you spend another 3 minutes to play with your own interface, but don't make too much noise. The drive, the file is in the folder, the channel here. Do you have trouble to unzip it?
No, I extracted and then after that what do you do?
Oh, you extract that, then you have to know where the file is, and you have to do the CD. In my case, I unzipped in the download folder. So if you change your current working folder to there, and you should be able to see those demonstration file right here. So that's what you need. Then you just run this. Yeah. Yeah, I can take a look at what you have over there. We have time. Yeah, so this for demo ICA. Yeah, you just run this. Just run. No, just run it. You should have that in. Yeah. Okay.
I see it. Then I see
Yeah. Okay, after that, make sure you will come back to the by default the working folder where you install the toolbox. That in my case, this is in my user documents and MATLAB. My toolbox was installed under this folder. Okay, and then clean your space, clear all, close all. Let's see, okay. Yeah, so we will switch to the download folder, and then you want to come back to this working folder by default. This is the place where I have toolbox installed. So I want to spend another 30 minutes to show you the spatial transcriptome toolbox. This is unfinished, so it's still have some issues when you click buttons. But you can give it a try and look at what's the main functionality in there. You need to click this button here, okay, to visit a GitHub page. So this is called STGA toolbox. It was SC for single cell. This is a spatial transcriptome. Very similar structure. You need to copy this quick installation code by just click this, copy, and then you have to paste this into MATLAB, and MATLAB will download this package and unzip it, and also add it to the parse. That's all. So I show you what I'm doing here. So I click this, copy these 2 lines of code, and come back to here, make sure I have single cell toolbox. And now I paste this to install the single spatial transcriptome toolbox. So after that, I should have SC and ST GA toolbox under the same folder. Yeah, so this is what the folder looks like at end after the installation. You had SC before already, and now ST, so side by side. Now, if you type STGA2, okay, this should give you the interface. And you can load this example data processed. Yeah, so it's this one here. Just let me know whether you have get this picture here. Oh, you have a different picture? Oh, okay, yes. Yeah, so if you load this, you get that. But if you use example data, you get, yeah, either way, yeah. Yeah, you just click OK, yeah. So process, so this is example data I had already prepared for you to show the tissue and also those gene expression spots. Yeah.
So what would you use this for? Like what analysis could you do with this?
Yeah, that's a good question. So you can look at the marker, the genes, if you look at this button here to look at the genes expression in different locations.
Okay.
Yeah, you select genes. So those buttons you can click, but some of those should not work, and because it's still under development. But major, yeah, that's good. Yeah, it just takes some time to load. It's fine. Yeah.
So the raw data for this sequencing will be the cell count, like
The,
matrix
yeah.
plus the location, right?
Yeah. That's right. Yeah, so what you can do with this is to first look at their corresponding location. You have the slide down there, so you can take off the spots and look at this tissue slide image, and then put it back and align them, and side by side you can start to look at those correspondence. You can put that into a 3D, so you can rotate and see what's the corresponding positions. You can change the size of those spots. I rotate. Yeah, in this resolution, I want to increase the size of those bubbles. I can click this to change. I can change the types and also different color map. Okay. Those very basic features. So another quick function you can try is to look at the gene's expression. So if you click this button, which the button under this big eye here, you should have a list of genes. So I gave a list of genes. And then you should be able to see those genes expression in the different spots. Yeah, I just select too many genes. Some of those genes, like this, is highly expressed. So you found this gene expression in almost all the spots, right? Some of this will be more sparse. Gray spot means there's no expression of this gene in that particular spot. And this gene may have some structure information. You see it's only expressed across this boundary here, okay? So put them together, you should have some idea about the cell type or the likely structure of enrichment of some gene expression in different locations. Okay, those ones on my screen, they are randomly selected. So many of them maybe just no information, but you can see, okay, some of this will tell you what's in that spots and why they cluster in this region rather than that region. That's because the tissue is different under the hood. So click this button to close all those small windows. Yeah.
Can you just upload the regular data like you would for the SCGDA tool and it would do that, or does it have to be in a specific format?
It had to be specific format. You have a data structure very similar to single-cell RNA-seq, the matrix with the genes. But also you have to have all this, not the cells, but this location of those spots.
Okay, and regular, like, the regular output doesn't have that, usually.
That's right. You have to have the data which you do the experiment to get that information. Yeah.
Kind of going off of that, can you show where the histology picture would be in the file?
Yeah, I will show you using example data set, okay? The one you can use is this one here. If you copy this and Google search, this will lead you to the geo database where you can take a look what the data, input data looks like for this kind of analysis. We should have a lot in the geo database, okay? So this sample is from a mouse colon, and this particular strain of mice. Yeah. Say that again. Okay. Yeah. Yeah. You can shut down your computer, not the MATLAB, and restart the MATLAB. Yeah. Sometimes you have run into this kind of a graphic, low-level graphic function issues. Yeah. normal bulk. Yeah, this is called a pseudo bulk analysis. Yeah, it's certainly possible. You can just pull these 50 cells and take average or summarize them into one sample. Yeah, yeah. I strongly against those kind of analysis because the variability is so important. You take average, you actually cheat yourself and get rid of them. Yeah. So for spatial transcriptome data, it looks like this. Okay, you still get this feature matrix that where you have expression of the matrix for the genes and not the cells, but the spots. Then you have this high resolution image. So if you download and unzip, you will see the image. That's all. You have the location, the position information. Okay? So this is a valid record for 10x Visium spatial transcriptome analysis. So what we are going to try is to use this ID. Okay, we are going to come back to this toolbox. I close this example toolbox, and I will start from scratch. So I will download that. In here, I can download using a session number. So the number I just copied. You can copy it from the channel in the Teams. And then you put that number in here, okay? And then just click OK. This data will be extracted, download from the website, load into this tool. But they're not a process. We have to do something for it. Mm-hmm. Yeah, so let's see. Yeah, so in this case, probably just shut down. Not even this? No. Oh, okay. So it's still busy? No.
Is there error?
Mm-hmm. Yeah. Yeah, I don't know. Even not shut down. Yeah, maybe you want to restart the machine even. Yeah. Yeah, so after you download this data, it's not aligned with your tissue image. So we have to do something, and in order to match this. So they all, you click this and manually turn around this and try to align them. So you can turn this and zoom in, zoom out, and you click M, this M to hold, to move this. Sometimes I have trouble to see how can I align them, so I don't know in this case. You have to find the right spots. But after you align them properly, you can save the next time you have aligned the picture. So in this case, I probably have to do this image and then click M, this button here, and then maybe flip again. Have to find the right. Yeah, it's not easy. It's not easy. I don't know. This is too complicated for this. Some others will be easier. You rotate. You use this to do the rotation, and use this to do the flip. Anyway, just assume I already done this, and then you can zoom out a little bit, stretch a little bit. So then you can start to look at the genes. If you know which gene will be important, then you directly point to that gene. I just randomly select a few and see whether it makes sense. There are quite a lot of zero genes. You can do a QC, okay, to get rid of those spots. So this is there's the genes, delete some of the genes which expression is too low. So it's very similar to a single cell RNA-seq, but here it's not the cells, but it's just spots, single spots analysis. You get rid of so many genes, only have 9,000 genes, which expression level is high enough. And then what I can do is to brush. For example, I can brush the region, and I want to look at a cell highly expressed in this region. So after brush those, you can select marker genes for this spot. And then top 10, it should have a list of genes highly expressed in that location compared to other regions. Yeah, so this is one I found. It's highly expressed, relatively high in this region I selected. This is one, okay. Another one. So in this way, you start to explore those feature genes, and especially if you know the structure and where to look at, you can start to understand these genes. It's highly expressed here. Yeah, I think that's all I want to share, and just let you know there are tools like this existing. If you have spatial transcribed data, you can load that in there. And also, if you have you want to reconstruct other people's study, if they released this spatial transcribed data in geodatabase, you can download and try. Play with them. Okay, it's time for questions or comments. This picture you can download from the geodatabase. This picture is here. Yeah. This format of data you generate from the 10x genomics. If you have that experiment, you will get this data. It will be processed with yes, it's a spatial transcriptome cell ranger. Yeah. Yeah. Yeah, so this is called the 10x VSUM. You have to use this to generate that. Mhm. This is not cheap, but now the resolution become higher and higher, and this year is like a spatial transcriptome year, and so many people, so many labs want to use this technology. It's very expensive. Um, so,
We did 4 samples, like 200, 28K or something. So 7 or 8,000 per sample.
yeah. So the data analysis will be more important because once you have spent such amount of resource to get this, you want to extract valuable information out of it. But now I don't know how to do it in reality.
Oh, the book can do it.
You can do clustering. You can cluster the spots, and based on so you can do something like that. And it will show you the cluster membership of those spots. Mm-hmm. Yeah, I mean, we know how to manipulate those data, but we don't know how to go deeper to understand the high level of biology directly out of this. Yeah. So we know how to do this basic annotation analysis of those data, but what I mean, we don't know how to analyze this for deep understanding of what's go next after you this superficial common pipelines. That's the topic that many people try to know what to do with this, basically. Yeah.
Is there any way, like for selector tools, that instead of a box, it could be like a lasso?
Select tools, yeah.
When you select like cells?
Select cells, yeah. The lasso will be a good way to find cells or gene. Yeah. Oh, you mean lasso, the selection of the region. Yeah. Yeah, what we can do is, if you want to select those spots, you can click with the shift key and select them. You know. Yeah, you can select with the shift key. Yeah, I show you this cluster results. If you using existing, so if you cluster your spots into here 5 different clusters, this is just like a single cell data that you color your different clusters, and they're automatically and show the location of those clusters. So all the cells, all the spots in this cluster, they group together in that region. And all the spots at this boundary has been clustered together, not using the spatial information, but just use the profile of the gene expression. And then you look at where they are located.
Oh, that makes sense. So
Yeah.
then using the lasso, like.
Yeah, you select the cells. Yeah, same clusters. yeah, if you really want to select those by hand, you can. You should be able to brush the spots, then click the shift, brush another spot. So in this way, you can select, but it's difficult to. For example, I'm selecting those cells, but they are different locations to make one cluster. It's possible. Yeah. All right. Okay. Lunchtime, everybody, and see you next week.